Okay, let's get started. Hello, my name is Yoichi Araki from Johns Hopkins University, working with Dr. Rick Hugnier. And title of my presentation is Shingapan isoform differentially regulate synaptic plasticity and dendritic development. As you know, genes are differentially transcribed to messenger RNA, which we call the alternative splicing, and that messenger RNA is translated to the protein to exert the distinct functions. In SHINGAP1, it is reported that 4 distinct C-terminal isoform was expressed in the brain. In SHINGAP1-related intellectual disability, many gene mutations were reported so far in SHINGAP1 gene. Importantly, these mutations affect all SHINGAP isoforms, both in messenger RNA and protein, to change its function. So it is critical to understand each isoform expression pattern and functions to understand SHINGAP1 related intellectual disability. This information will provide the mechanisms of the disease and will lead to the efficient gene therapy by unique one SHINGAP isoforms. This is showing the protein structure of SHINGAP1. You can see last cap domain in the center, and C-tail has a communicative phosphorylation site and coil domain, which is required for phase separation of with PSE95. Near C-terminus, there is alternative splicing junction points. Beta is coming from the alternative splicing of exon 17 and 18. Alpha 1, alpha 2, and gamma isoforms are coming from the alternative splicing of exon 18, 19, 20 junctions. To characterize the SHINGAP isoform expression and localizations, we raised each individual antibodies to specifically recognize each SHINGAP1 isoform. We saw most of the isoforms are specifically expressed in the brain, and also this region-specific Western blotting shows almost all isoforms are enriched in the cerebral cortex and the hippocampus in the adult brain. We also characterize a temporal expression of SHINGAP1 isoforms. As you can see, beta and gamma isoforms were expressed early compared to alpha 1 and alpha 2 isoforms, and this expression is replaced with alpha 1 and alpha 2 expressions later. We calculated the composition of SHINGAP1 isoforms. As you can see, most of well-studied alpha 1 expression comprise only 25 to 35% of SHINGAP1, highlighting the importance of study all isoform to elucidate SHINGAP1-related intellectual disability. Again, beta is well expressed around the birth, and this is replaced with alpha 1 and alpha 2 expressions later. We previously reported that SHINGAP alpha 1 isoform is interacting with PSE95 to form condensed phase in synapses to provide a physical basis of postsynaptic density. So we investigated if the other SHINGAP isoforms have similar functions in the cells. We transfected each SHINGAP1 isoforms with or without PSE95 in hex cell and fractionated into soluble fraction and a pellet phase-separated condensed fractions. This is a graph showing how much protein is going to phase-separated condensed phase in the cells. As you can see, when I expressed SHINGAP alpha 1 with PSE95 here, most of the proteins are going to the condensed pellet phase, reconfirming the previous observation in 2016. Interestingly, the other isoform, especially beta isoforms, are more going to the soluble fractions and less going to the condensed phase, suggesting the distinct biochemical properties of SHINGAP1 isoform in the cells. Reflecting these distinct biochemical properties in the cultured cells, the in vivo brain localization of SYNGAP1 isoform are also distinct between SYNGAP isoforms. We fractionated the mouse brain into S2 cytosolic fraction and postsynaptic density PSD biochemically. As you can see, SYNGAP isoforms are enriched in PSD fractions. Among these, alpha 1 isoform is going to PSD fractions mostly among SYNGAP1 isoforms. Interestingly, reflecting the soluble biochemical properties of beta isoform in the cells, beta isoform is going to cytoplasmic soluble fractions in the brain in vivo compared to the other isoforms. This is a quantification. Beta isoform is actually 30 times more going to cytoplasmic fraction compared to the alpha 1 isoform, suggesting the special role of this isoform outside the synapses. We surprised that, in contrast, extremely low existence of SYNGAP alpha 1 isoforms in cytoplasmic fraction. This extremely low ratio is much lower than any other famous postsynaptic density molecules such as PSE95 and AMPA receptors. SYNGAP is a last gap protein where this hydrolyzes last bound GTP to become a GDP, and this makes RAS inactive. And it is also reported SYNGAP has gap activity for other small g proteins such as LAP1 and RAC1. So we wanted to know the specificity of each SYNGAP isoforms to inactivate these small g proteins. We co-expressed SYNGAP and small g proteins and how much active GTP-bound small g proteins are decreased by SYNGAP isoform expressions. For example, active LAP1, as you can see here, is decreased by alpha 1 expression, but you can see more dramatic decrease in beta expressions. This is a quantification here. You can see the beta has generally maximal gap activity towards all small g proteins we assayed, including RAS and LAP and RAC. It is interesting that alpha 1 isoform has relatively low gap activity but has preference towards RAS gap. We previously reported that SYNGAP and PSE95 are postsynapses at basal state, as you can see here. But once NMDA receptor and CAMK2 was activated, CAMK2 phosphorylates SYNGAP, and this dissociates SYNGAP from PSE95, inducing the rapid SYNGAP dispersion from the synapses. And we found this dispersion is essential for AMPA receptor insertion and the synaptic enlargement during LTP. So we wanted to know if the other SYNGAP isoform has similar functions for synaptic plasticity process. We expressed AMPA receptors and EBCHERI, who are the marker for the synaptic morphology. You can see the AMPA receptor is inserted and spines are enlarged after chemo-ATP inductions. For example, you see very small spine here, but this is enlarged after chemo-ATP. You see larger spine here compared to the basal here. And also, you can see rarely AMPA receptors at the basal state, but you see the AMPA receptors after chemo-ATP. And you can see in quantification, spines are enlarged after chemo-ATP and AMPA receptors are getting inserted after LTP. And as previously reported, these spine enlargement and AMPA receptor insertions are both occluded when I knocked down SYNGAP by shRNA, shown here, suggesting that the essential role of SYNGAP1 was synaptic plasticity. And we rescued this phenotype by expressing each individual SYNGAP1 isoforms. Interestingly, alpha-1 isoform is successfully rescuing this LTP occlusion. Alpha-2 marginally rescued this, but all the other isoforms cannot clearly rescue the plasticity deficits. This indicates the exclusive role of alpha-1 for the synaptic plasticity. We noticed that when we knock down SYNGAP very early in the development at DIV3, the dendritic development is severely impaired in the culture neuron. So we would like to know which isoform plays this neural morphology role. This is a graph for the show analysis, how much dendritic urbanizations were observed at the distal segment. As you can see, you can partially rescue this phenotype by expressing alpha-1, alpha-2, gamma isoforms, but only beta isoform perfectly rescue the dendritic development deficits upon SYNGAP1 knockdown, suggesting the exclusive role of SYNGAP beta on the dendritic urbanization at early development. In conclusion, we characterize a variety of functions and biochemical properties of SYNGAP isoforms. We see different temporal expression of SYNGAP isoforms where the beta expressed early, alpha-1 and alpha-2 is replaced when they become adults. SYNGAP beta has maximal gap activity and expressed early and has exclusive functions on the dendritic development. SYNGAP alpha-1 is phase separated with PSE95 in postsynaptic density and has exclusive role on the synaptic plasticity. These informations are critical for understanding SYNGAP-related intellectual disability, how these isoforms contribute to disturbance of the synaptic connection as well as neural circuit formation. I would like to thank you all the people in the project in Dr. Rick Hugnier's laboratory, as well as the collaborators. Thank you so much for your attention.